the remote sensing technician at, we call it FSF, the Field Spectroscopy Facility. We are a lending library of spectroscopic instrumentation based at the University of Edinburgh. So we loan sensors to academics and researchers to use for free. If that's something that you're interested in or you want to learn more, I've tagged our website at the bottom there, so feel free to contact us. But today I'm going to be talking about how to apply field spectrometer data to rank wheat genotypes by drought resilience. How do I switch? Great. So this data was collected as part of a Pangeos cost action hosted by the Norwegian University of Life Sciences in June of 2025. So the purpose was to demonstrate the capabilities of various remote sensing and plant phenotyping sensors and collect data for Project Smart Wheat, which focuses on how we understand drought response in wheat. So this project, the PI is Dr. Zoe Halem, who was meant to give you a second presentation today, where she was going to discuss how we can integrate different sensors, so not only field spectrometers, to find key traits that indicate drought tolerance in wheat, but again, she will reschedule that talk. A bit of background about the project. So Smart Wheat is aiming to characterize drought response in Norwegian wheat varieties on plant growth and physiology. The aim is to develop drought resilient wheat varieties and later simulate their drought response through AI modeling. A bit of background as to why. So we know that climate change is increasing the frequency and severity of drought events, and that poses a major threat to global food security. Wheat is one of the world's most important cereal crops, and its productivity is becoming more and more vulnerable because of intensifying drought stress. We also know that drought affects different wheat cultivars in different ways. So their strategies to maintain photosynthetic activity can vary. So some of them improve their water use efficiency, other ones trigger their photoprotective mechanisms. And we also know that the timing relative to the plant's life cycle and when that stress event or that drought event happens can have a different effect on the plant. And so that leads us to this question of, can we use spectral data to assess drought resilience in different wheat varieties? To do this data collect, we used a polytunnel enclosed environment with 6 wheat genotypes, and they had 2 treatments, so drought stress and optimal watering. The wheat varieties were both, some were drought tolerant and some were drought sensitive, which allowed us to do a really comprehensive evaluation of the sensor's performance given the different stress tolerance mechanisms. Remote sensing, and specifically field spectroscopy, has been shown to provide insights into the effects that stress has on plants. So here we used an SVC field spectrometer, which is a hyperspectral sensor that measures over the 350 to 2,500 nanometer wavelength range. And so that's really crucial because it's measuring into the shortwave infrared, and that's been shown to tell us about water stress in plants. We had to pre-process the data and actually had to remove the water band. So water absorption regions are notoriously very noisy in field spectrometer data. And so even though we are looking at water or trying to look for differences in water content, we have to remove those as they're going to introduce potentially spectral artifacts and can't really show us consistent spectral information. But luckily, we can focus on the shortwave infrared as well as calculating spectral indices to tell us about water content. We then wanted to link the spectral response with the plant's physiology. So we measured some agronomic variables, specifically NPQ or non-photochemical quenching. So this is the plant's photoprotective response to stress. If there's a drought event or a stress event, the plant's going to absorb excess light energy and dissipate that as heat. And that prevents it from getting damaged. So essentially, if we've got a stress event, we're expecting higher NPQ values. We also looked at quantum yield, or QY, which is a parameter for assessing the maximum photochemical efficiency of photosystem 2. This is another indicator of plant health under stress. So under stress conditions, we'd expect the QY value to decrease as photosynthetic efficiency is declining. And lastly, we just compared it to yield, which is the overall productivity indicator. Again, like quantum yield, we expect that reduced photosynthesis is going to lead to a decrease in overall biomass. So some preliminary results from this. Here we're just looking at the drought response per variety. So the water band regions are just around like 1300 and 1800 nanometers. Those are the really noisy regions. But here we're taking an average of all of the control spectra for each variety and the average of the drought spectra for each variety and subtracting to get a different spectra. And ultimately what we're seeing here is there's no kind of consistent trends. There's no systematic behavior of the drought tolerant or the drought sensitive varieties. They've each got quite a heterogeneous response, which could be potentially caused from them just being heterogeneous in their genetic makeup. We then decided to do some statistical analysis. So here is a PCA plot of the average of each plot measured with the field spectrometer. And again, we're not really seeing any sort of consistent clustering. We know that PCA is a unsupervised dimensionality reduction technique, and it doesn't consider the group's structure. So potentially it's capturing variance that's unrelated to drought stress. So there are some interesting features in here, specifically if you look at demonstrant, these squares, the control and drought plots are quite well clustered, which is what we'd expect given that demonstrant is meant to be drought tolerant. So the drought plots shouldn't be looking very different spectrally than the control ones, but that behavior is not really consistent among all the varieties. We then did some heat maps looking at linear discriminant analysis and some spectral indices and comparing those to the physiological traits or the agronomic variables, quantum yield, NPQ, and yield. And if we first look at LDA, specifically LD1, we're seeing a negative correlation, relatively weak, but still negative with NPQ, and then quite a strong positive correlation with quantum yield and yield. And so we know that LDA is a supervised classification technique, so it already knows to separate out the control and the drought plots, and it takes those predefined groups and maximizes that class separation. So it's showing us that lower stress, this negative correlation with NPQ is linked with increased photosynthetic efficiency and higher yield. So this positive value for quantum yield, an indicator of plant health, is likely telling us that under non-stressed or control conditions, that quantum yield value should be increasing, and it should be showing us that inverse relationship with NPQ. And then LD2, there was relatively weak correlations among all three agronomic variables. Then if we look at the spectral indices and compare those to the physiological traits, if we focus specifically on quantum yield and yield first, we see positive correlations with NDWI, WI, and WBI. These are our water indices. So this is indicating that higher plant water content is supporting higher photosynthetic efficiency and productivity. And then on the other hand, the drought-related indices, MSI, which is the moisture stress index, and NMDI, which is the normalized multiband drought index, I think, are showing negative correlations with quantum yield and yield. And this is just exactly what we'd expect, that under drought conditions, there's going to be reduced water content and reduced productivity. And then finally, if we just look at that middle column, the correlation between NPQ and the spectral indices, we're not really seeing any sort of strong correlations with any of the spectral indices. That could be caused by NPQ just having responding more variably to stress, or potentially we needed to do more than one data collect over the plant's life cycle to see any sort of relationship with NPQ, or potentially NPQ is not a reliable indicator of stress in this context, but we're not really sure about that yet. Then to kind of visualize all of that on a PCA biplot, we have the water-related indices, NDWI, WI, and WBI, pointing in the same direction as yield and quantum yield. And those are kind of plotting generally with the control group. And then in the complete opposite direction, we have the drought-related indices. And then NPQ is like not really plotting with or corresponding to anything at all. Again, what we saw with the correlation heat maps. So yeah, finally, I just wanted to point out some key wavelength regions that we were able to identify from spectral analysis. So if we look at first and second derivative spectra, as well as investigating the PCA loadings and the LDA structural coefficients, we're able to pick out some key regions that can then be later compared to other sensors. So specifically the green peak around 550 nanometers, telling us about pigment changes, as well as the vegetation red edge, telling us about photosynthetic activity. Then we have the near infrared, which indicates changes in overall canopy structure. And finally, the shortwave infrared, telling us about plant water content, especially we can see super high values in the PCA loadings and the LDA structural coefficients in the shortwave. Finally, some conclusions from all this. Across all of the analytical approaches, we observed consistent relationships between spectral features and agronomic performance. Plant water content is the primary driver of both spectral response and plant productivity. Hyperspectral measurements can effectively capture drought-driven variations in wheat, and overall these results are demonstrating that field spectroscopy is a valuable component in multi-sensor phenotyping workflows for identifying drought-resilient wheat genotypes. Okay, thanks.